Hello everyone, welcome to today's coffee seminar. I'm a senior research fellow in coffee in the Centre for Crop Science, and I'm very pleased to introduce today's seminar by Araz Solman. But first, I would like to acknowledge the traditional owners and their custodianship of the lands on which we meet today. On behalf of the traditional owners, I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. And also, if I can just do a few housekeeping things, the seminar will go from 12 o'clock today till 1 p.m. And at the conclusion of the seminar, we will have a question and answer session, but please start typing your questions into the question and answer section as soon as you can think of them. That way we'll be ready to answer them at the end of Araz's talk. So for now, as I said, I'm very honoured to introduce today's speakers, Dr. Araz Solman. Araz was awarded a PhD from Curtin University in 2020, working on pathogen interactions in co-infected wheat plants. And before that, he came to the University of Western Australia from ICARDA to investigate the use of antitransparents to increase drought resistance in wheat for his master's degree under Kadambot Siddiq and Ken Flower. For the last 14 months, Araz was working as a plant pathologist at the Department of Agriculture and Fisheries, based at the Hermitage Research Facility in Warwick. And he has been a very valued member of our research community here at Hermitage. And he has made great contributions to summer crop pathology, especially in mungbeans. But unfortunately, funding for his position has run out, and we regret that Araz will be moving on and is currently looking for a new position. In fact, this is to his last day, which is quite sad. The title of Araz's talk is A Tale of Two Crop Diseases. And if that sounds like something out of 1001 Nights, it might be because Araz is originally from the Middle East. He's told me that the name Araz is the name of a river in Azerbaijan and is as common as John among Kurdish people. So, welcome, Araz, to your seminar.
Thank you, Bob, for your nice introduction. I really appreciate that. And it's been a nice 18 months that I spent at Hermitage Research Facility, and I'm really excited to have this opportunity to present on my last day at work and to have the honor to be able to present some of the work that I did back at Curtin University, and also showcase the work that I've been doing within mungbean at Hermitage Research Facility. So today is all about plant pathology, and it's about a specific area of plant pathology where the infection or the disease is a result of more than one pathogen, or what I would call throughout the talk a co-infection. And I have slightly adjusted the title of the talk to include the word double trouble, a tale of two crop diseases. And I'm sure you're wondering why it's double trouble. But if you stick around throughout the slide, I'm sure you will be able to answer this question. And if I fail to answer that question throughout my slides, please ask me at the end why it's double trouble. So let's dive in. What's on the menu today? I'll give you some background introduction on what's co-infection and why should we bother about co-infection, or the infection that happens as a result of more than one disease. We will look at the current knowledge or the current understanding of co-infection and what we know about co-infection. And then we will progress into three different sections of this presentation. The first one has a methodology that we developed or was developed for this specific co-infection. And then we will go into a field survey of co-infection to show how co-infection is common in West Australian wheat belt. And then we will look at detailed in vitro and in-plant co-inoculation experiments. And then finally, we'll finish with my current work that I do in mungbeans to show that co-infection can occur in other crops, including mungbean. So yeah, there's something for everyone. There's methodology, there's field survey with complex field data, and there's also detailed in vitro and in-plant with transformation, GFP and RFP. So there's, please stay tuned, there's everything for everyone. There's something for everyone. So why should we care about co-infection, and what is co-infection even to start with? For 50 years or so, we've been looking at plant pathology in a simplified reductionist approach, where the disease, we've been considering disease as a result of a single host and a single pathogen or a single disease interaction. And this has been amazing because we've made so many major discoveries, and we know so much now about how plants interact with their host and with the pathogen, and how pathogen infect their host, and how the plants defend themselves against invading pathogens. But this reductionist simplified approach of a single host and a single disease does not mimic what happens in nature. In nature and in agriculture and agroecosystem, plants often get co-infected with more than one disease. And it's in fact very rare to see a plant with a single disease in the field. And this complex interaction of more than one disease often display properties that we cannot capture by simply looking at each component separately. So this is the whole idea of today's talk. It's looking at how hosts in the field can be co-infected and how that would have a major impact on the consequences for the disease. So what do we know about co-infection? We don't really know a lot about co-infection, and most of our understanding of co-infection comes from the theoretical work that was done on high order animal and like human, and also social insects like honeybees. But I've put together a cartoonish way to show what would be expected when we have more than one pathogen in a system. So in this example, if we had, let's say we have a gray and orange pathogen, and the gray and orange pathogen both sharing a host. And if the gray and the orange pathogen have a different research acquisition strategy, say the orange pathogen is more efficient at acquiring host resources, then over time, we would expect the density of the orange pathogen to go up on the expenses of the gray pathogen in a typical case of resource competition. So both pathogens coexist, but density of the orange pathogen will go higher up. What if the orange pathogen has the ability to produce toxins like lots of phytopathogenic fungi? Then we would expect the population of the orange pathogen to sharply increase and that will cause a significant decline in the population density of the gray pathogen. And that leads to a typical case of toxin aggression or chemical aggression. What if the plant has the ability to target one pathogen or another? Then if the plant targets the orange pathogen, the gray pathogen, we would expect the population of the gray pathogen to decline, and that will give a chance for the orange pathogen to go up. And if the plant targets the orange pathogen, then vice versa. But if the plant has the ability and an efficient mechanism to target both pathogens, then the whole scenario can change. There are also possibilities where the diseases can arrive at different time or invade different spaces or niches within the host. Say, for example, a disease land on a leaf and another disease infect the root system. Then these two pathogens will have minimum overlap, and their population density can increase or decrease depending on the condition favoring one or another. And if both pathogens arrive at different times, say the first one arrived early in the season and the last one mid-season or late season, then we would expect minimum overlap and minimum competition. So this is what's likely to happen when two pathogens enter into a relationship or an interaction in terms of the density. But what happened to the disease? What happened to the host? When we have more than one pathogen invading the host, their combined damage could either be additive, so 1 + 1 = 2, or or non-additive, and that could be synergistic or antagonistic. There's also a chance that both diseases land on a leaf, and the leaf is a limited space and limited resources, and these two pathogens can enter into competition, leading to what's so-called the tragedy of the common, and the ultimate ultimately for the benefit of the plant. There are also scenarios where the first arriving pathogen can get access to host resources and space, and in this case, we will have a first come, first served scenario. But if the first arriving pathogen elicit host responses that are negatively that can negatively impact the later arriving pathogen, then in this case, the host will be primed for further attack. So these are the questions that haven't been answered in the space of co-infection, and throughout the next slides, I'll be attempting to answer some of these questions using a three-way pathosystem, a very unique three-way pathosystem that consists of wheat as a host and two fungal species. So the first fungal species in this three-way system is known globally as tan spot, but here is known as yellow spot or yellow leaf spot. In the US, it's known as red smudge, and it's caused by a fungal pathogen known as Ptr. But for the sake of today's talk, I'll call tan spot or Ptr, I'll call them Ptr or Ptr. The second pathogen that was involved in the three-way system is Septoria odorum blotch. So it's a fungal disease. It's also known as gloom blotch. And gloom blotch or Septoria odorum blotch is caused by a fungal pathogen known as Parastagonosporum odorum. But again, the fancy name for today's talk, I'll call it Pan. So we have, in the three-way system, we have wheat as a host, Ptr and Pan. And why Ptr and Pan are a good example of co-infection? Well, it's very simple. Ptr and Pan both infect leaves, and they reduce the green leaf area and hence photosynthesis and yield. They're very common, and they're stubble-borne diseases, so we'd expect to see them commonly in wheat or wheat-grown crops. They have this polycyclic nature of disease, so they produce multiple cycles of the infection throughout the season. So we'd expect to see them throughout the season with no differences in arrival time. It's also thought that Ptr and Pan can co-occur, but we do not have data to back this up. The only evidence that we have that support that Ptr and Pan might co-occur is that both Ptr and Pan share a very famous and a common toxin known as toxate. And there's a very famous work by Timothy Friesen and his colleagues that put together a hypothesis that Ptr acquired toxate from Pan through a long-term commitment of horizontal gene transfer. So this is the only information that we have, or the only evidence that we have that Ptr and Pan might co-occur. And when we go to the field and look at Ptr and Pan, or look at tan spot and Septoria odorum blotch, their symptoms can overlap. They look slightly similar or very similar. And here in this slide, we have tan spot on the left or on the right, and tan spot has this classic tan necrotic lesion with a yellow halo around it. And Septoria odorum have similar halo, but the lesion can be irregular in shape. But the problem is, when we have them side by side, Ptr and Pan, it's really hard to tell them apart. So the symptom overlaps so much, and if they next to each other, you wouldn't be able to tell them apart, and that created a problem. So if I had gone to the field and start looking at them and visually scoring them, I would be making so many mistakes, and also assuming that I can tell them apart, I'll only be able to tell apart what's on the leaf, what I can see externally, and also I would not be able to tell the distribution or the contribution of each pathogen to the total disease load. Instead, we can use qPCR, we can use quantitative PCR, and we know quantitative PCR can do the job because it relies on the DNA. And the other tick for quantitative PCR is it's quantitative, so we can get information on the total disease load in addition to the distribution or the contribution of each pathogen. And it also gives us information on the full load, so that's what's inside and outside the leaf. So that's why we relied for this research, I relied on using qPCR, and the qPCR methodology took very straightforward process. Primers sequences of all known Ptr and Pan isolates were aligned, and in this case we included lots of isolates because we knew the aim is to take this to the field and see how common Ptr and Pan co-infection is in the wheat belt. So we aligned all known sequences of Ptr and Pan. We identified region of interest, which we targeted to design DNA primers, and then to multiplex the assay and to be able to detect and quantify Ptr and Pan at the same time, we added hydrolysis probe to be able to multiply and also to increase the specificity of the assay. So here's some results. The first thing we did was testing our primers. So we did an endpoint PCR. I did an endpoint PCR where we had 6 repeats of the Ptr DNA or Ptr DNA and 6 repeats of Pan's DNA. And then we run this assay, we run this PCR, and our primers were able to detect Ptr and Pan DNA. Despite that, our primers were very small. So they're very, very unique. They're very, very similar in size, but one was 112 and the other one was 108 or 104. So they were very similar in size, but our DNA primers were able to detect Ptr and Pan. And again, because this was meant to go to the field, so it was important to test that our assay does not cross-react to other species or does not cross-react to wheat DNA. So we put, in a 2-repeat assay, we put a DNA from 5 different fungal pathogen or fungal species that thought to co-occur with Ptr and Pan or thought to co-occur in the wheat belt. And we tested our primers, and again, Ptr primer detected Ptr only, no cross-reaction with other pathogen or wheat DNA, and Pan primers detected Pan only, no cross-reaction. So it's all good. And we had the primer tested. The next step was to multiplex the assay and put the probes in. So we did a qPCR this time with, again, Ptr and Pan in the assay, in each assay, and with the other 5 fungal DNAs or fungal species and wheat DNA. And again, we're looking at the relative fluorescence against the cycle number on the qPCR machine, and the amplification of the Ptr started at 15, no cross amplification with other fungal species or wheat DNA. And similarly for Pan primers and probes, they started around 15 or 16 cycle, and no cross reaction with other DNAs from other pathogen or wheat DNA. The other thing I did was test the recovery of fungal DNA by changing the ratio of Ptr and Pan. And the reason I did this is because in the field, we don't know what's the proportion of these 2 pathogens, and we don't know what ratio they might be on. So we created matrices of DNA where we change the ratio of Ptr to Pan from 1:1 down to 1 to 10,000 or up to 1 to 10,000, and then vice versa. So when we had the DNA ratio 1:1 all the way to 1 to 1,000 of both pathogen, the assay was reliably able to detect, to recover the DNA that we put in the assay. So there was good recovery of fungal DNA, good quantification of fungal DNA. But when we had the ratio of one pathogen down to 10,000, that's when the assay was unable to detect that pathogen in very low quantity. So that happens in both Ptr and Pan. We were unable to detect the pathogen that was in very, very low density in the assay. Then we did the same ratio assay, but this time we increased the background DNA of wheat DNA. So we did this because we don't know what's the level of infection that we will experience in the field. And this time we went from 1 to 1 to 1, so Ptr Pan to wheat DNA, down to 1 to 1 to 2,000 unit of wheat DNA. And as you can see, the influence of wheat DNA, background wheat DNA, is very minimal, up to ratio of 200 to 1. But when ratio went to 2,000 to 1, that's when some interference started to occur, and the assay was lagging by about 15 to 20%. So this is the limitation of the assay. If we see a ratio of 2,000 to 1, then the assay might give us less reliable results. But that's the best we could do. And the final step was to see whether we are able to tell and quantify Ptr and Pan and whether they occur or co-occur in the field. So we went and collected leaf samples from a farm site and brought back a few samples, and the samples typically shows symptoms of tan spot. So there was no, there's only the necrotic lesion with a yellow halo around it. So there's no any evidence of Septoria nidorum or Pan. All we can see is tan spot. But when we stick our leaves on a qPCR machine and analyze them using the quantitative PCR, things look very different. So here we're looking at diseased leaf area, the relationship between diseased leaf area from 0 to 45, and fungal DNA in nanograms for 50 nanogram diseased leaf DNA. So as we can see, Ptr DNA is in abundance, and that's highlighted in the blue dots. And that Ptr DNA corresponds very well with the total DNA load, or the total disease load. But there's also Pan DNA or Septoria nidorum DNA, and that exists in a very low quantity. But the visual scoring would not have told us this information. Only the DNA was able to tell us that there is very low quantity of nidorum DNA. So what's the conclusion of this section? As I said, the reductionist approach might be old slightly, and it does not really mimic what happens in the field. In the field, often we see pathogen in combination, more than one disease and more than one species. And the good thing is we have the technical capacity now to be able to address these complex questions of co-infection systems. The second section of this talk involves a field survey in the wheat belt, and the objective of the survey was to see how common Ptr and Pan co-infection is in the Western Australian wheat belt. And we also were interested to see if the Ptr and PAN ratio that we saw in the last slide changes by the variety or the cultivar and cultivar site or the canopy level. And the whole rationale for doing this is because we have programs that address Ptr and Pan separately. But if these pathogens co-occur, then it makes so much sense to target these pathogens as a complex. So this is the whole rationale behind doing this job, is if they co-occur, we might be able to find ways to better manage them as a complex. So what we did, I went to the wheat belt of West Australia, and I collected I visited all these sites that are shown on the map, and I brought back about 432 samples. So I went on to all these sites, collected leaf samples. These leaf samples were grouped by the variety. So we took information from the farmers about the variety and what they grew. We also grouped these leaves by the canopy level. So whether it was taken from the first leaf or second leaf or third leaf or from flag leaf. And we also grouped these leaves by the growth stage of the plant. Was the plant on zealot growth 12 or 15 or so on? And before I take the leaf from the plant, I score the disease visually. So me and a bunch of other people scored the leaf visually, and then we stick these leaves in tubes and send them back to the lab and do DNA extraction for quantification of fungal biomass. The leaf mainly fall into 3 different categories or 3 different cultivars. One of them is Yitpi, and the other 2 were Emu-Rocco and Magenta. Yitpi has a very susceptible reaction to Ptr, but an inverse or moderately resistant interaction with Pan. Emu-Rocco has a moderately resistant response to Ptr, but very susceptible to Pan, and Magenta is moderately resistant to both diseases. So we had more varieties, and in the paper you will see the full analysis of all the varieties, and we call Yitpi, Emu-Rocco, Magenta, I think maize and others, but I'm using these as an example. So we analyzed these samples for fungal DNA, and we came up with this complex table. So the leaves were analyzed for DNA, and the first measure of disease load that we calculated from these leaves was disease incidence. So this is a measure of how many leaves detected positive for either or both fungal DNAs. And as you can see, disease incidence was very high. 91% of the samples that we collected from the wheat belt were infected by either or both pathogens. And the second analysis, or the second piece of information that we can get from the qPCR analysis, was the pathogen prevalence. So of these infected samples of these samples that tested positive for fungal DNA, which disease we have prevalent in these samples. So what you see here is the presence in percent, and the number in parentheses, the number of samples that were tested. And on the left side are the sites. And as you can see, the prevalence of samples that were co-infected by both Ptr and Pan were 94%. So 94% of the samples that we collected from the wheat belt of West Australia had Ptr and Pan in combination. Only less than 6% of samples had Ptr only or Ptr only, and less than 1% showed DNA of Pan. So yeah, co-infection is very, very high by these 2 pathogens, and there is a strong evidence that Pan does not even occur on its own as a pathogen. We always see it in co-infection with Ptr or Ptr. We also looked at the visual scores. So we had collected visual scores in the field. So we scored the plants and scored the leaves, and that goes from 0 to 100 on the y-axis. So this is visual scores, and we're looking at the visual scores of 2 leaves, the upper leaves in the canopy and the lower leaves in the canopy of 3 different sites, Minganoo, Muresk, and Wagen. So this is north, midwest, and west. And we had the three genotypes, Emu-Rocco, Magenta, and Yitpi, for the 3 developmental or 3 growth stages, 24 to 26, all the way to 55, from 55 to 62. So that's anthesis, all the way in anthesis. So as we expected, the level of infection, the level of disease goes up as we progress, as the plant progresses throughout the season. And there's a lot more disease on the lower leaves compared to the upper leaves. And this is just simple because the lower leaves are under a lot more pressure, disease pressure. But when we do exactly the same, but instead of looking at visual score, we look at fungal biomass, we see a completely different picture. So we see clear site differences. So Minganoo is a lot more infected than Wagen and Muresk, especially than Wagen. And also, we don't see that clear increase in fungal DNA in Muresk and Wagen. But what is clear is there's an increase in fungal DNA as the season progresses, and there's a lot more DNA on lower leaf compared to the upper leaves. Now, which disease was present at different times? So this is the important information. Which pathogen was contributing to the infection at different times, or which pathogen was contributing to the total fungal load? And here we're looking at a complex four-way interaction. So we have on the y-axis, we have disease score, or visual disease score. On the x-axis, we have fungal DNA. And we have the growth stages, 24, 26, all the way to 62. So these are the growth stages. And we have the upper leaves and the lower leaves. And we have fungal DNA of Ptr is represented by the black dot, and fungal DNA or the relationship between fungal DNA and the score of Pan is represented by the light gray dots. And as you can see, early in the season, Ptr is a dominant disease on both upper and lower leaves, especially on the lower leaves. And that continues on the lower leaves. In the mid-season, we still see Ptr, but Pan slightly increase here and becomes also important on the upper leaves. And Pan does not become an important disease until the end of the season. And when the plants were at enthesis, both Ptr and Pan were nearly contributing equally to the co-infection. So we have this dynamic of the abundance of these two diseases changes by season and leaf position, especially at the end of the season. Both diseases become equally important. So how well our assay did, our qPCR assay did for quantifying fungal DNA at different times. And now we're looking at the relationship between fungal spores, fungal DNA, and visual scores of the three growth stages, so 24, 26, all the way to 62. So we're essentially looking at early, mid, and late season of all the leaf and genotype combinations. And as you can see, early in the season and late season, the relationship between fungal DNA and visual scores does not fully explain what we see. So the DNA or the visual score does not explain fully what we see, but that relationship becomes a lot stronger mid-season when the R-square explains nearly 70% of the variance. There's a perfect explanation for this. We think early in the season, both diseases, or at least Ptr, is still progressing, and what we quantify is what we see is what's on the leaf on the top. There's a lot more that we don't see within the leaf, and late season this relationship becomes poor because the interference of environmental factors becomes high. For example, senescence and herbicide application or fungicide application or other things that can interfere with what we can see exactly in the field. But mid-season, when the pathogens both at their peak, especially Ptr at this time, Pan would start the season at this time, that relationship becomes a lot more strong. So what's the conclusion of this section is Ptr and Pan co-infection in the wheat belt is very common, and we can give a number now and say nearly more than 90% of the samples that we collected from the wheat belt contains DNA of Ptr and Pan. Ptr and Pan abundance is dynamic throughout the season. So we think, and as we saw in the data, Ptr start the season and continues throughout, while Pan starts mid-season and continue to the end of the season. And we also can see that qPCR is a good fit for doing scores, especially when we analyze our data in mid-season, especially when we collect our sample in the mid-season. So this is the conclusion of the second section of this talk, but I've left the more interesting aspect of this interaction, which is looking at the mechanism of the interaction. How how PTR and PAN interact and what regulates that interaction, what's the fate of the infection when we have both of them at the same time, and also when both of them arrive at different times. And also, what are the mechanisms of this interaction? So all these questions, we wanted to address them in the next slides. And the first thing we did, we started very simple by simply looking at Ptr and Pan on plates. So we put Ptr and Pan agar plates, and we had Ptr and Pan in the middle of the plate with no distance between them. And we have plates with Ptr only, and we had plates with Pan only. And we measured growth 3 days and 6 days after we plated the pathogens. And as you can see, Ptr can rapidly take over Pan. And by 6 days after doing the plating, Ptr had completely taken up the whole plate, significantly growing a lot more than when it's on its own or when Pan on its own. So when we have Ptr and Pan on the same plates, their growth just goes wild, a lot more than when Ptr on its own or Pan on its own. And much of this growth corresponds to Ptr. So PAN growth remains restricted in the middle of the colony and does not grow beyond that middle center. Then we did Ptr and Pan and on distal end of the plate. So we put Pan and Pan on either end and Ptr on either end. And the reason we did this was to see if there is self-recognition of these two pathogens. And as you can see, by 3 days, the growth is very limited, but by 6 days, Ptr and Pan, when they're on separately, so when we have Pan and Pan, the growth is limited, Ptr and Ptr growth is limited, but when we put Ptr and Pan, the growth of Ptr becomes significantly more than Pan, and they start creating this exclusion zone of the interaction between the two diseases. So yes, they are able to interact, and I've looked at this area in where the arrows are, and I've looked at that area microscopically. Their fungal growth can intermingle; they can grow into each other, but they do show this clear exclusion zone. The other thing I did was transforming the Ptr and Pan with a green fluorescent protein. So one with a green fluorescent protein and the other one with a red fluorescent protein. And the aim was to do a complete implant and in vitro analysis to see how they interact. And here we're looking at wild type Ptr next to wild type Pan and wild type Ptr next to GFP transformed Pan. And as you can see, the GFP transformed Pan is not majorly restricted. It's still okay. There was loss in fungal spore production, some loss in fungal spore production, but the growth was not inhibited. And when we transform both and put them on the leaf, yes, and then again, their combined damage when we have them both on a leaf, their combined damage is significantly greater than when we have Pan on its own or Ptr on its own in the wild type. So this was done to make sure that we haven't, by the transformation, we haven't changed their growth or we haven't altered their ability to cause infection. And as we saw, they were both able to cause disease, and they were both still able to cause significantly more disease on the plant, on the detached leaf assay than when they were on their own separately. Then we stick these slides under the microscope. So we realized three different zones. So we call this Ptr zone in the area where Ptr grows freely with no interaction over resources. And this is the Pan zone, but the area in the middle, we call them the interaction zone, our confrontation zone, let's call it. So as you would expect, Ptr produce Ptr fungi. So we didn't see Pan on this area. These are typical Ptr hyphae, and these are typical Pan hyphae. So nothing exciting. on the Ptr zone, we see Ptr or Ptr. On the Pan zone, we see Pan. And then when we look at them transformed, we see Ptr again glowing with a fluorescent tagged protein here, or fluorescent staining here, and Pan with a GFP just looking green. So this is exactly what we expected, nothing interesting. And then when we look at the confrontation zone in the middle, we see that hyphae can intermingle and they can form networks, and they have no issues of these two fungi to join the hyphae, absolutely no issue. So we would not expect interference because the hyphae can intermingle. And then when I do a Ptr and Pan in a live imaging under the confocal microscope. So here we're looking at Ptr in the red, so transformed with RFP, and Pan transformed with a GFP. And this is a video, but I've cut this from a video. What you can see here is Ptr and Pan can co-occur, and there's absolutely no exclusion in their growth. So you could see these light orange. So this is an area where Ptr and Pan can even grow over each other with overlap, and there's no exclusion of one or either one. Then we progressed into detached leaf assay. So this time we infected plants with Ptr, Pan, or their co-infection. So we had Ptr, Pan, and we had treatments where we had both Ptr and Pan in combination. So here we're looking at four different genotypes: Emuroc, Helbid, Magenta, Yitpi, and this is the corresponding infection treatment. And underneath, we're looking at qPCR results. So Ptr and Pan did not produce any symptoms by 3 days post-inoculation, but when we had Ptr and Pan in the same as a co-infection treatment, plant rapidly showed symptoms by 3 days post-inoculation, and that's consistent with nearly all genotypes except Magenta. So in all the 3 susceptible genotypes, Emuroc, Helbid, and Yitpi, we had very quick symptoms when we had Ptr and Pan in combination. But when we put these leaves, when we stick these leaves 3 days after inoculation in the qPCR machine, what we detect is 100% Pan, so Ptr. So there's only Ptr at this stage causing disease. Pan was under the detection limit on the qPCR machine, and there was no detection of Pan in all of the genotypes. So yeah, Ptr and Pan co-inoculation caused quick symptoms on detached leaf, and much of that quick symptoms correspond to Ptr DNA. So here we're looking at days until symptoms appeared on the plant for Pan, Ptr and Pan and Ptr co-inoculation of 4 genotypes. And as you can see, it took about 7 to 8 days on most genotypes to get symptoms, with the exception of Magenta. So 7 to 8 to get symptoms of Pan. Ptr about 6 to 7 or 6 to 8 on most genotypes. But when we had Ptr and Pan as a co-infection, that dropped to 3 to 4 days, or on average 3 days. So by 3 days, plants that were co-infected by Ptr and Pan showed symptoms, while Ptr infected plants took about 7 days and Pan took about 8 days. And by 12 days post-inoculation, or by 12 days post-inoculation, these are the Pan-infected plants, and these are the Ptr-infected leaves, and the Ptr Pan co-infected leaves were significantly more diseased compared to each pathogen in isolation. Now, what if we play with the arrival sequence of Ptr and Pan? So we change one over the other one by delaying the inoculation of the other one by 24, 48, and 72 hours, and vice versa. So we started with Ptr Pan together and delaying the Ptr by 24 to 48, 72, and vice versa, starting with the Ptr and then delaying the inoculation of the other pathogen by a day 2 and 3. So what happened to the disease symptoms, or how quick the symptoms expressed? And there was this magic. When we had Ptr and Pan together, the symptoms appeared about 2 to 3 days or 2 to 4 days in all genotypes. And we had symptoms very quickly when Pan was inoculated 1 day and Ptr inoculated 24 hours later. The reverse order, so the reverse order when we had Ptr first and then Pan 24 hours later, did not show the same results. And that was consistent in all genotypes, all varieties. So the magic is Ptr, Pan in combination at the same time, quick results or quick disease and rapid infection, and there's also rapid infection when we have Pan first and Ptr a day after, but the reverse did not cause the same results, did not show the same results. Now we're looking at disease score, so 0 to 8 of the 4 genotypes. And when we have them infected by Pan, Ptr, the orange, and green, Ptr and Pan at the same time. And in all genotypes, even in the most resistant genotype, magenta, when we had both genotype, the expression of disease was very quick and also was progressively higher than the other 2 genotypes throughout the course of the experiment. So it went from 6 to 15 days of scoring, so every 3 days. And we always had a lot more disease on the leaves that were co-infected than the leaves that were infected by Pan or Ptr. Now, which pathogen was in action when we analyze these leaves, when we analyze exactly the same as this result, but break down this green bar into its component? So we're looking at fungal DNA on the y-axis in picograms, and these are the days post-inoculation, and we have the 4 genotypes of the 4 varieties. And in the light blue is Pan as a single inoculation, Ptr in the light darker blue, Ptr single inoculation, and then in the light green, Pan co-inoculation, and dark green, Ptr co-inoculation. And what we can see is consistently Ptr is the dominant pathogen in the co-infection, and in the green bar, when Ptr with Pan its growth is a lot higher, significantly higher than when it's Ptr on its own in the blue bar, in the dark blue bar, and that was consistent in all genotypes. But what happened with Pan in the co-inoculation? Well, Pan, as it looks here, it was just inhibited and its growth was nearly close to 0, but there was some detection of Pan, but significant detection were for Ptr. So yes, Ptr benefits from being co-inoculated with Pan, and that probably corresponds to heart disease. Now, what happens when we have the sequence changed? When we change the sequence of the co-infection, what happens to fungal DNA? So what happens inside the leaf? When we have Ptr and Pan simultaneously, or Ptr first and Pan delayed from 24 to 72 hours, and the reverse, Pan first and then Ptr delayed from 24 to 72 hours for the genotype Emuroc or variety Emuroc, Halbert, and Yitpi. And here we're looking at blue bars for Ptr and orange bar for Ptr and blue bars for Pan. And as you can see, when we have Ptr and Pan simultaneously, or when we started with Ptr and delayed the inoculation of Pan, it's always Ptr. So together, Ptr. Starting with Ptr, we always see Ptr, okay, or Ptr. So there's first come, first serve. But what happened when we delay Ptr? So when we start the inoculation with Pan and delay Ptr, here's where it becomes interesting. So when we delay Ptr by 24 hours, that gives some chance for Pan to express itself, and there's some DNA. Delay a bit more, and Pan has a chance. Delay a lot more by 3 days. Pan nearly dominates the co-inoculation. So there's a typical first come, first serve, and that might be explained by resources, availability of space, and so on, and also even can be explained by the effector presence and the effector interaction. When we have currently work under review about the effector interaction. So yeah, first come, first serve, PTR start first, or when it's simultaneously, PTR takes over. But when we delay PTR, that gives a chance for Pan to express and become important. And when we stick transform PTR and transform Pan under the microscope, here's what we see. So we're looking at fluorescent microscopy of Ptr. So this is Ptr inoculating detached leaf assay of wheat of the Helbert, of the genotype of variety Helbert. And as you can see, Ptr enters wheat leaf mainly through the stomata. So we repeated this inoculation, I think 50 times, or 50 attempted inoculation, attempted penetration were almost exclusively through the stomata. And the same can be seen from the scanning electron microscope. PTR enters, so put the padding, push padding against the stomata and into the stomata of the plant. And yeah, this is another view of the stomata underneath in the green. So that's PTR infection peg, and that's the stomata, leaf stomata, wheat stomata. But when we put Pan only, Pan does not penetrate the leaf through the stomata, and instead Pan takes the approach of directly penetrating the leaf through the cuticle. So Pan creates something called infection peg, and that infection peg gives the pathogen the physical ability to penetrate the leaf and enter inside the plants. So there's difference in their ability to, in the way they enter the plant, by PTR dominantly through the stomata, entering through the stomata, while Pan dominantly entering through the cuticle. And when we look at their infection over time, what we see on the leaf, so this is a confocal microscopy of transformed Ptr, and what we can see is Ptr always, after it enters the plant, remains on the leaf surface. So when we look at the leaves, when we look at dig deep inside at a different confocal depth, we only see Ptr at a very shallow depth of the leaf, so mainly in the epidermal tissues. What happened with Pan? Well, Pan enters really deep inside the vascular bundle. So we don't see Pan until we dig deep down in about 40 confocal depth. That's when you see Pan. So there's not only differences in the way they enter the plant, there's also difference in where they reside within the plant. So Pan mainly remain in the surface, while, PTR mainly remains in the surface. Pan enters deep inside the vascular bundle. And when we do an assay where we do leaf sectioning at intervals, at fixed intervals, so we put here in the middle, we put Ptr and Pan inoculum on a detached leaf, and then we section that leaf 1 centimeter apart, 1, 2, 3 on either ends. So what happens here? In the middle and on the sides, PTR grows all the way to the end of the leaf. So on both sides, you see DNA, Ptr's hyphae. But when you come in the middle, you see a stack of Ptr hyphae in the blue and only tiny bit of Pan hyphae or Pan mycelia in a dense stack or trapped in a dense Ptr mycelium. So when we have them in the middle, Ptr has the ability to grow beyond the infection site, but Pan gets stuck in a dense Ptr mycelia and cannot progress beyond that infection site. And that's why we think Ptr can overtake Pan, because it can trap it within a dense mycelium. But there's no exclusion. So what's the conclusion? Ptr can outgrow Pan in vitro and in planta, but there is no antagonism, so they can coexist and co-occur, no problems. Their co-inoculation seemed to lead to rapid and more disease, and often that benefits Ptr density. So Ptr takes over Pan when they co-inoculated and rapidly takes over Pan and often cause more disease than when Ptr or Ptr on its own. Time matters when we have Ptr and Pan, and Ptr always benefit when it's first come, first served. But when we delay Ptr inoculation and give Pan the chance to infect, Pan can take over. Ptr and Pan have ability to enter the plants using different mechanism or different spaces. So stomata versus direct penetration of cuticle. So hence, and they can also reside within different leaf tissue, and hence we think there's a space differentiation or there's a niche differentiation that enables their coexistence. But when we have Ptr and Pan in the exact same spot, Ptr does not give chance to Pan, and Pan get trapped within dense Ptr mycelia and does not progress beyond the infection site. So this concludes the work that I did back in Perth at Curtin University, and in the next few slides, I want to show you what I've been doing with mungbeans, and just to show you that co-infection does occur in other plants, including mungbeans. So in my work here at the Department of Agriculture at Hermitage, I looked at 3 different diseases: halo blight, tan spot, and powdery mildew. So of mungbeans. So halo blight of mungbeans is caused by a bacterial disease that causes necrotic spot with their own halo. Tan spot is caused by also a bacterial disease, but often causes irregular shaped necrosis with a tan halo, and that's hence called tan. And powdery mildew is a typical talcum powder symptoms caused by the fungi. But when we go to the field and take any leaf sample or infected leaves, when we take infected leaf samples of mungbean, what we see is this is a leaf that has 3 different diseases. So this is halo blight showing symptoms. So in the red arrows, that's tan spot, and this is powdery mildew. So despite that the 3 diseases have a different lifestyle and different infection mechanism, the 3 diseases can co-occur, supporting that co-infection does co-occur in many different plants. And when we attempt to breed plants to increase their susceptibility or increase their resistance to certain disease, we see that typical inverse relationship between powdery mildew and halo blight or tan spot. So when we select for resistant to powdery mildew, that often correspond to susceptibility to halo blight and vice versa. So there's this inverse relationship between mildew and mildew being a biotroph and halo blight or tan spot being a typical necrotroph or follows nearly necrotrophic lifestyle. And this is very simple because mildew requires green leaf tissue to extract nutrients, while halo blight kills tissue to extract nutrients, hence the relationship is inverse. So these are the people that I acknowledge from the work that I did at the Center for Crop Disease Management in Curtin University. So Richard, Oliver, Mark, Fran, and Caroline. And the superstar of most of the work that I do is John Hamlin from UWA, from University of Western Australia. And these are the people that I work with, that I've been working with at Hermitage Research Facility. So it's my last day, and it's very excited that I've had this opportunity to present on my last day at work. But please, my commitment to this and to plant pathology continues. And if you have any question, if you have anything, if you want to get in touch, this is my private Gmail and this is my phone number. Please get in touch, and you'll find me always open to new possibilities and happy to collaborate. Thank you very much.
Well, thank you very much, Araz, for this interesting talk. I think we're just about out of time for questions. So I might spend the time just concluding from today, thanking everyone for attending. And also you will get an email if you're on the coffee seminar list, you will be sent an email, and please fill in the poll at the end so that we can know how to improve or what other topics you would like to see in the coffee seminars series. If you haven't signed up for the newsletter, for the email list, you can do that at coffee.uq.edu.au/science seminars, so that you get sent an alert each time we have a seminar. And next week's seminar will be by Associate Professor Jim Hannon. So his topic will be fractals, plants, insects, and orchards. Also a very interesting scientific title. So reflections on life with L systems. So we look forward to have you join for that seminar. And thanks again, Araz. All the best for your future. It's been a privilege having you here, and I wish you all the very best.
Thanks a lot, Bob. Really appreciate it.